continuation of the previous area, how this cuticle is made, or the biosynthesis of chitin in particular. Chitin is biosynthesized by the insect epidermis. The active chitin monomers are secreted into the extracellular apodicient spaces where they are polymerized to form chain by the enzyme chitin synthetase. Chitin synthesis in insects is controlled by 20-hydroxyecdazone and it is pH and temperature dependent chemical process. Chitin synthesis takes place via a metabolic pathway of 8 or more steps, converting hemonymph trehalose into chitin as follows. Like trehalose, with the help of trehalase, turns into glucose. Then with the help of hexokinase, it turns into glucose 6-phosphate. Then glucose phosphate isomerase turns that into fructose 6-phosphate. Then glutamine fructose 6-phosphate aminotransferase. We have to glutamine and glutamic acid is also involved there. Glutamine changes into glutamic acid. So this fructose 6-phosphate turns into glucosamine 6-phosphate. Then acetyl-CoA turns into CoA, and with the help of glucosamine 6-phosphate N-acetyl transferase, this glucosamine 6-phosphate turns into N-acetyl glucosamine 6-phosphate. Then this N-acetyl glucosamine 6-phosphate turns into N-acetyl glucosamine 1-phosphate with the help of phosphoacetyl glucosamine mutase. Then uridine diphosphate N-acetyl glucosamine pyrophosphatase turns UTP into PP, which will eventually transform N-acetyl glucosamine 1-phosphate into UDP phosphate N-acetyl glucosamine. Then chitin synthetase UDP to chitin N-acetyl glucosamine. Chitin microfibrils, chitin poly beta 1,4 N-acetyl D-glucosamine. Microfibrils are ranging from 25 to 30 angstrom in diameter. It is present in cuticle in crystalline form. Each crystallite consists of 60 to 21 chains forming 3 sheets, each containing 6 or 7 chains. Crystallographically, chitin is heterogeneous, consisting 3 polymorphs like alpha, beta, and gamma chitins. All 3 polymorphs occur in insects as microfibrils. The viscoelastic polymers. Alpha chitin, it is common one. It consists of an orthorhombic unit cell with 4.74 angstrom in the crystallographic alpha axis, then 18.86 angstrom in the B axis, and 10.30 angstrom in the C axis. The unit cell is composed of disaccharide sections of 2 antiparallel chains bounded in 2 ways, intramolecular hydrogen bond and another bond between successive chains forming sheets along the A axis. The CH2OH group on one side of the chains forms an intramolecular hydrogen bond with the carbonyl group of the next residue, while the CH2OH group of the other chain forms an intermolecular bond. Various amide groups take part in hydrogen bonding. Alpha chitin chain is rigid in constitution. Beta chitin. It is rarely found in insect cuticle, mostly in cocoons of some beetles. It consists of a monoclinic unit cell with 4.84 angstrom in the crystallographic alpha at A axis, 9.26 angstrom in the B axis, and 10.38 angstrom in the C or fiber axis. The unit cell is composed of a repeating disaccharide. The chains are arranged in parallel fashion. The C double bond OH single bond N hydrogen bonds form the sheets. The CH2OH groups are hydrogen bonded to the carbonyl groups of the next chain along the alpha A axis. There is no intermolecular hydrogen bonding between the sheets. Then gamma chitin, it is found in cocoons of some beetles and peritrophic membrane of some insects. The unit cell measures 4.8 angstrom in crystallographic alpha axis, 29 angstrom in the beta axis, beta or B axis, and 10.3 angstrom in the C or fiber axis. The biaxial dimension of beta chitin is about half of the alpha chitin, while that of gamma chitin is 3 times larger than that of the beta chitin, and thus alpha chitin molecules folds back on itself. In other properties, it is similar to beta chitin. A series of lamellae, helicoidal model, screw carpet model, Blackwell and Weiss model. Different types of models. These are some chitin microfibrillar arrangement. Different models are there in chitin microfibrillar arrangement, like Bouligand's helicoidal model, Bouligand helicoidal model. Bouligand's helicoidal model. It asserts that the microfibrils occur in sheets within the cuticle, where all microfibrils are parallel to one another and are also parallel to the surface plane of cuticle. Neighboring sheets of microfibrils are rotated with one another in the same direction but with variable or fixed angle of rotation. A group of sheets of chitin microfibrils are thus helicoidally arranged in the protein matrix. So it asserts that the microfibrils occur in sheets within the cuticle, where all microfibrils are parallel to one another and are also parallel to the surface plane of cuticle. Neighboring sheets of microfibrils are rotated with one another in the same direction but with variable or fixed angle of rotation. A group of sheets of chitin microfibrils are thus helicoidally arranged in the protein matrix. Then Weiss-Foulkes screw carpet model. It asserts that the microfibrils are continuously secreted and sheets of microfibrils change their position vertically gradually, so that a microfibril present in the region of the helicoid turns slightly and contributes to more than one sheet of microfibrils. The screw carpet model is analogous to crystal structure in some synthetic plastics. Then Blackwell and Weiss model. Blackwell and Weiss proposed a model illustrating structure of chitin protein complex on the basis of X-ray diffraction studies. It shows that a supercoil of 15 to 30 chitin chains forms a core of the chitin protein complex. A chitin core is surrounded by a spherical protein sheath. The chitin fibrils forming core are perpendicular to the axis of the chitin protein complex fiber, while the protein subunits are arranged radially around the chitin core. The chitin protein complex fibers measure 7.25 nm in total diameter. The protein subunits are spaced around the chitin core in a 6⁻⁻1 helical exhibiting hexagonal arrangement with a fiber repeat at Ca 15.3 mm. Proteins. The proteins of cuticle are of three types: the arthropodin, resilin, and sclerotin. Arthropodin is a mixture of various proteins and is devoid of sulfur-containing amino acids. Arthropodin. I'm sorry, not anthropodin, it's arthropodin, arthropoda. It is arthropodin. It is a mixture of various proteins and is devoid of sulfur-containing amino acids. It resembles sericin or silk gelatin in its general properties. It is water soluble. It constitutes primarily the pro and endocuticle, representing the untanned protein. Then resilin. It is a rubber-like protein that occurs and evident as a colorless elastic cuticular ligament such as the wing hinge ligament, clipeolabral suture, spring, then tergosternal ligament, and at various sites of articulation. It is composed of about 60 amino acids which are arranged in the linear fashion, and formation of cross links is avoided to ensure elastic property of the protein chain. In the cuticle containing resilin, the pore channels are entirely lacking. It can stretch under tension and can return to its normal form as soon as the tension is over. And the sclerotin, it is also known as the tanned protein. It is brown or amber in color and occurs in the exocuticle. In fact, it is a tanned arthropodin produced due to the action of quinone with the N-terminal amino groups of the arthropodin molecule. And finally, the quinoid protein links into the other protein molecule by the cross-linkage. Then lime. In some aquatic insects, lime or CaSO3 is deposited and may form a major constituent of the exocuticle particularly in the diptera larvae and puparia. Then pigments. The insect exocuticle possesses various types of pigments such as melanin, carotenoids, and the proteins producing fluorescent coloration in the wing scales of butterflies. In most of the scarabid and cicindelid beetles, an entire epicuticle is covered with a thick layer of secretory material of the dermal glands and provides brilliant pigmentation. Cuticular modification. Next topic is cuticular modification. So we mentioned about structure and chemistry of this insect integument. Next, cuticular modifications. Part 2. Cuticular modifications. Macrotrichia. These are the hollow, hairy exocuticular processes. They are of great taxonomic importance as their distribution differs from species to species. There are following types: clothing hairs that cover the entire body and appendages as found in honeybees. Then glandular hairs occurring around the opening of various epidermal glands and can be seen in a few Lepidopteran larvae. Then sensory hairs are those constituting various mechano and thermoreceptors and are occurring on the antennae, mouthparts, legs, and various regions of body in all groups of insects. Clothing hairs, glandular hairs, sensory hairs are those constituting various mechano and chemoreceptors and are occurring on the antennae, mouthparts, legs, and various regions of body in all groups of insects. Chemoreceptor sensory hairs. The wing scales are modified and variously pigmented microplate-like structure that invade the wing surface of the adult butterflies and moths. So these all four, like clothing hairs, glandular hairs, sensory hairs, and the wing scales comes under macrotrichia. Then microtrichia are the fine sensory hairs without having the basal articulation. They are confined to the wings in some Mecopteran and dipteran flies. Cuticle, epicuticle, invagination, and muscle. Spines can be seen as the cuticular process in some insects, aquatic dragonfly larvae. Some adult beetles are possessing the cuticular horn on the apical region of the head, particularly in males. These structures represent the highly modified cuticular process and are of epidermal multicellular origin. Apodemes and apophyses. There are two types of cuticular invaginated processes. The hollow invaginations are called the apodemes and are the sites of muscle attachment. The solid invaginations are called the apophyses. These structures form the endophragmal skeleton inside the body of insects, which provide mechanical support to various organs and to the whole body. They form distinct skeletal structures such as tendorium in the head and endothorax in the thorax. Apodemes and apophyses. Cuticular membranes. In some regions of the integument, the exocuticle is discontinuous due to lack of tanning, and thus with such parts of cuticle remain membranous. These membranes are flexible and perform intersegmental intrinsic or extrinsic articulations according to their location. And ecdysial lines. Similarly, in case of larvae at various regions as head and thorax, the exocuticle is absent, forming the ecdysial lines. Due to the absence of exocuticle, the ecdysial lines becomes very weak along with the cuticle of exuviate spills at the time of ecdysis. Due to this reason, the ecdysial lines are also called the lines of weakness. Ecdysial lines. And next is about molting. All insects shed and replace the cuticle a number of times during post-embryonic development, and the process is known as the molting. Molting is a complex process involving first a separation of the old cuticle from the hypodermis, apolysis, followed by a shedding of the old cuticle, ecdysis. Immediately after ecdysis, tanning of cuticle, sclerotization take place. Apolysis. The apolysis starts with increase in the number of size of the epidermal cells of the hypodermis. Due to repeated mitotic divisions of the epidermal cells become tall, columnar in shape, and are closely packed. They exert marked tension at the cuticular surface and as a result separate themselves from the cuticle. Due to the separation of the hypodermic from the cuticle, formation of subcuticular space take place. The epidermal cells or modified molting glands discharge the molting fluid in subcuticular space. The molting fluid is a granular gelatinous fluid and contains chiefly two enzymes, the proteinase and chitinase, which are specifically digesting the endocuticle of old cuticle. Initially, the molting fluid is inactive, but as soon as the cuticular layer of a new cuticle is secreted, it becomes active. As the molting fluid digests the endocuticle of old cuticle, the subcuticular space increases gradually. On the other hand, the cuticulin layer moves forward with the synthesis of wax and polyphenol layers. After the formation of definite epicuticle, the successive layers of procuticle are deposited beneath, and the subcuticular space is gradually occupied by the newly synthesized cuticle. The molting fluid is capable of digesting almost all endocuticle of the old cuticle except some indigested material, which later persist in the form of the ecdysial membrane below the old cuticle. The inocytes secrete the cuticulin layers of new cuticle, which is impermeable to the molting fluid. The other epicuticular layers, the wax, polyphenol, and semen layers, are formed from the substances transported by the epidermal cells through the pore canals. The procuticle is also a product of epidermal cells, which is discharged via the pore channels. The semen layer in some insects is secreted by the dermal glands. It is deposited over the wax layer at the last, just before the ecdysis. In ecdysis, the insect body is enclosed with an old cuticle consisting of epi and exocuticle on a newly synthesized epi and procuticle. The old cuticle is known as the exuvia, and the developmental stage bearing exuvia is called the ferrate instar. Ferrate instar. The insect body is enclosed with an old cuticle consisting of epi and exocuticle on a newly synthesized epi and procuticle. The old cuticle is known as the exuvia, and the developmental stage appearing exuvia is called the ferrate instar. Due to digestion of old endocuticle, the ecdysial lines develop on various parts of the exuvia. Due to the muscular activities of the inner developing insect, the exuvia spills along the ecdysial lines. Besides the muscular activity, the insect swallows either air or water. As a result, the gut is distended, causing increase in hemolymph pressure. The blood is subsequently pumped from the abdomen into the thorax by muscular activity. Due to enhanced blood pressure, ultimately the thoracic old cuticle spills along the lines of weakness. After breaking up of exuvia along the ecdysial line, the new insta comes out with bringing out its head and thorax first, followed by the abdomen and appendages. Not only the integumental cuticle or exuvia, but all the old cuticular structures are cast off, including the endophageal skeleton, tracheal lining, and intima of fore and hind gut. The mechanism of ecdysis in the Cyclorhyphon is, however, much specific. The head of the fly bears a frontal reversible sac called the tilinum. P is silent. Tilinum. It is capable of expansion and contraction of the blood is pumped into the head from the thorax and abdomen and withdrawn alternatively, causing a periodic expansion and contraction of the tilinum, which spills off the cap of the puparium through which the fly finally emerges out. After emerging, the tilinum becomes non-reversible and its muscles undergo degeneration. In the old fly, it is represented by a tilinal suture. At the time of ecdysis, a new cuticle is very soft and milky white, providing no substantial mechanical support to the insect body. At this stage, the blood acts as a hydrostatic skeleton. And next is about sclerotization. Sclerotization or tanning is the last process of molting, which involves differentiation of the procuticle into outer hard exocuticle and inner soft endocuticle. The exocuticle represents the tanned part of the cuticle. It is simply a conversion of the protein arthropodin into sclerotin by the initiating formation of cross links between the adjacent protein molecules. The cross links are formed by quinones. The concentration of tyrosine increases highly in the hemolymph just before the ecdysis. Tyrosine is converted into dihydroxyphenylalanine dopamine to N-acetyl dopamine and variety of quinones. The N-acetyl dopamine diphenol is transported via pore canals into the outer part of the epicuticle, where it is oxidized to a quinone by an enzyme phenol oxidase. The quinone diffuses across the epicuticle into the outer part of procuticle, and after forming cross links along the protein molecule produces the exocuticle. Quinone always forms links with terminal amino groups of the protein molecule having dibasic amino acid like lysine. It results in the formation of catechol-like protein, which is later on oxidized to a quinoid protein. The quinonoid protein links to another protein molecule. As a result of tanning, the chitin protein molecules are closely packed due to which the cuticle of the body wall becomes thinner, hard, sturdy, and dark. The darkening of cuticle seems to be a result of sclerotization as well as polymerization of excess quinones into melanin. The bursicon hormone, secreted by the cerebral neurosecretory cells in sarcophaga, controls the process of sclerotization, where ecdysone plays a key role in controlling various molting events. After sclerotization, darkening of cuticle occurs in large number of insects. It results, no doubt, initially from a process of sclerotization, but due to polymerization of excess quinone molecules, leading to formation of melanin pigment also. Various pathways of melanin synthesis are shown in figure. Tyrosine, dopamine, NADA, NPAD, like that. So we just covered this portion also. Thank you for learning with me.